Now we can start. I'm sharing my slides.
Yes, sir.
Slide is visible.
हाँ सर।
Yes, sir.
Yeah. Uh, we can start from what is genome. Uh, in your second semester, we can start from what is genome. Uh, if you go to genome, a genome is an organism's complete set of DNA that comprising of nuclear and mitochondrial DNA, both nuclear and mitochondrial DNA inside the cells. DNA is a total hereditary material present in an organism. Hereditary material means it transfer um, uh, fathers to its offspring. That is hereditary, and it may be DNA or RNA. This hereditary material can be a DNA or a RNA. Then, if we go to if we go to the genome, then genome is a totality of chromosome. Uh, that unique to a particular organism or any cell within the organism. Genome present within a cell and inside the chromosomes, and each genome contain all of the information that needed to build and maintain that organism. All the genomes uh, contains all the information. What is the information part? That is coding for RNA or proteins, which build and maintain that organism. Then how genome and genotype differs? Then genome is a total hereditary material, as we discussed. It transfer from fathers to father and mothers to offspring, and this genotype information content within the chromosome. This genome is present within the chromosome. Now, that chromosome is the small portion of the genome is a small portion of the chromosome. If we go to the then we discuss about the chromosome, and this is the chromosome, and the small portion of this chromosome is called as genome. Then if we go to the genetic organization, how this genome is organized in the cells, each DNA molecule associate, this is the DNA molecules, and this DNA molecule associated with different proteins molecules. The DNA have molecules which attach with the protein molecules. That protein is called as histone proteins, which called this DNA. And this DNA molecule and its associated protein is called as chromosome. These DNA molecules folded to form a chromosome, and this chromosome is arranged such a manner to form the organization of the DNA. And this organization holds true for prokaryotic and as well as eukaryotic cells. This chromosome present in both the prokaryotics and eukaryotic cells. If we go to the types of genomes, then there are four categories of genome according to different kinds of organism. If we go to the prokaryotic means pro means primitive. Prokaryotic genome, karyons means nucleus. Then if we go to the prokaryotic genome, then the prokaryotic genome is double-stranded DNA. Genome contain double-stranded DNA, and this is circular in nature. The DNA is like this, circular in nature, and having one chromosome and a nuclear structure. What is nuclear structure? This is a irregular organization of genome. We can discuss further. Then if we go to the second class, that is eukaryotic genome. Eukaryotic genome also contain this double stranded DNA, and this is linear. This is like this linear form, and here in prokaryotic, that is circular form. And this eukaryotic genome contains many chromosomes. Like for an example, in human beings we have 23 pairs of chromosomes. uh, then it contains a nucleus. Nucleus is a organelle which contains this chromosome inside the nucleolus. But this is a nucleoid structure. Nucleoid means irregular organization of genomes. Then if we go to the organelle genome, third category is organelle genome. That is peculiar characteristics for chloroplast and mitochondria. Chloroplast in plant and mitochondria in different animals. This is also a hereditary material. This is basically transferred from mothers to the child. Then if we go to the virus, this is important. Virus contain both single-stranded and double-stranded DNA and RNA, and this packed under glycoprotein complex. Like if we for an example, if we know about the SARS-CoV virus, this contains spike proteins, that is glycoprotein above the surface of this virus. This is crown like structure. These glycoproteins are crown like structure. This as you know, this virus genome contains both single stranded as well as double stranded DNA or RNA. Then how this genome is packaging inside the cells. If we go to the packaging of DNA into chromosome, how this DNA is packaged inside these cells, uh, like chromosome. If we go to the packaging of DNA into chromosome, serves several important functions. Chromosome is compact form of DNA. This is not a compact form. DNA is not a compact form. Chromosome is a compact form of DNA. How this form is happen, we will further discuss. Then it protects the DNA from the damage. If we compress one structure, then it will protect from other damage, like different stress like factors may damage our DNA. Then this compaction happens after this DNA is supercoiled. Then only packaged DNA can be transmitted efficiently to both daughter cells when cell divides. Then these packaged DNA are divided to form this chromosome to form chromatids. Then it divides to chromatids. Then these chromatids are efficient to both daughter cells when the cells is divided. This division takes place during this meiosis process. If we then go to the organization, how this organization is takes place in viral, prokaryotic, and eukaryotic cells. If we go to the genome organization of prokaryotes, as we discussed in your previous slide, the DNA is in nucleared form. Nucleared form means irregular form. If we go to the genome organization in prokaryotes, they have small bodies and small genomes. Thus, prokaryotic cell is very small and they have small body and the genome is very small. They do not contain a nucleus, they do not have a nucleus and any other membrane bound organelle. In prokaryotic, there is not any found of membrane bound organelles, and these are small in size, uh, small and circular DNA present inside the nuclear region. As we discussed, nuclear means the DNA is organized in irregular shape. They have single chromosome that floats in the cytoplasm. As we discussed, it do not contain nucleus, so the DNA is floats inside the cytoplasm. The genome size ranges in between 10 raised to 4 to 10 raised to 7 base pairs with a high gene density. They have base pair 10 raised to 4 base pairs, means as we know DNA contains 4 bases A, T, G, C, and if we go to the RNA, then RNA contains A, U, G, C. So this genome contains 10 raised to 4 to 10 raised to 7 base pairs with a high gene density. They have high gene content. Apart from this single chromosome, some bacteria have extra chromosomal DNA called plasmid. If you go to the little structure here, green-like structure, this is called a single chromosome, so extra chromosomal DNA, that is called plasmid DNA. If we go to the structure of plasmid, then prokaryotic also have frequently carry one or more smaller independent extra chromosomal DNA called plasmid. If you go to the structure of extra chromosomal DNA, the plasmid is look like this, circular in circular form. Plasmids are not genomic DNA. They are not genomic DNA. They are extra chromosomal DNA outside the chromosomes, and uh, they are accessory DNA molecules and small circular DNA molecules that have ability to self-regulate. These plasmid DNA have ability of self-replications, means doubling of one plasmid to two plasmid, increase the copy number. Replicates means increase the copy numbers. Unlike the larger chromosomal DNA, plasmids typically are not essential for bacterial growth. This DNA is not necessary for bacterial growth. Only chromosomal DNA is necessary for this bacterial growth. Then what is the importance of plasmid? Then plasmid provide advantage to bacteria such as antibiotic resistance, herbicide resistance. This plasmid is only important for the antibiotic resistance in bacteria. As we know about the antibiotic resistance, this plasmid creates antibiotic resistance in bacteria. In addition, unlike chromosomal DNA, plasmid are often present in many complete copies per cell. Then, addition to this chromosomal DNA, this plasmid DNA also present inside the bacterial cell. Then, if we go to the for an example, E. coli, Escherichia coli, that is E. coli, go to the bacterial genome, then bacterial chromosomal DNA is usually a circular molecule that is few million nucleotides in length. They have smaller circular DNA, which has is the few million nucleotides, that is 4.6 million base pairs in E. coli, that is 4 points in Haemophilus influenza, that is 1.8 million base pair. This causes this uh, bacteria causes flu-like symptoms. This is called Haemophilus influenza. Then a typically bacterial chromosome contains a few thousands of different genes. This contains a few thousands of different genes which encodes for their structural uh, part of the bacteria, and this structural gene sequence account for majority of the bacterial DNA. The non-transcribed DNA between adjacent genes are termed intergenic region. There is as we can discuss about further this intergenic and intragenic regions of this DNA. Then, if we go to the packaging of this DNA, then prokaryotic cells typically have smaller genomes. They need to compact their DNA is still substantial. If you go to the example of E. coli, then E. coli must pack in 1 millimeter chromosome. Length is 1 millimeter, but that is uh, packed to form a 1 micrometer in length, and that uh, compact inside the cell, and it is less clear how this prokaryotic DNA is compacted. Then if you go to this DNA, the circular DNA, then it forms a loop domain, loop-like structure, and this loop-like structure forms the DNA into a compact manner. And these loop-like structure are formed with the help of these DNA-associated proteins here in prokaryotics, and we can discuss about in eukaryotics, that is histones molecules. Then if we go to the looped chromosomal DNA, then it form a supercoiling using different enzymes. Then we discuss about these enzymes, and this supercoiled and loop like DNA to form a plectonic supercoil form. If with the different type like structure, one is plectonic supercoil form, the in toroidal supercoil form, and the different forms I like, and it forms a compact like structure, and it fit inside the prokaryotic cells. And here the supercoiling occur by different DNA topoisomerase. This topoisomerase causes unwinding, replication, rewinding. Unwinding means during replication process, this topoisomerase unwind to forms like this circular form, because in these circular forms DNA can replicate it, not like this form. In this form DNA cannot replicate it, so unwinding is required. For this unwinding, DNA topoisomerase is required. Then we go to rewinding. Rewinding means after unwinding, after DNA replication, rewinding occur to form a this supercoil and looped DNA. Then, for example, a topoisomerase called DNA gyrase catalyzes negative supercoiling of the circular DNA found in bacteria, and this DNA uncoiled and relaxed in order for a DNA polymerase to bind for DNA replication and RNA polymerase to bind transcription. This uncoiled, after this uncoiling, DNA polymerase bind for replication. Means if DNA polymerase is bind, then it's called replication. Replication means copy number increases, and transcription means if RNA polymerase bind, it transcribes to mRNA, messenger RNA, for coding sequence for synthesis of proteins. Then if we go to the genome organization in eukaryotes, then eukaryotic genome is a linear and conforms the Watson-Crick double helix structural model. As we know, Watson and Crick first described the double helix structure of DNA. For this, they have taken Nobel Prize in 1954, and this embedded nucleosome complex. This nucleosome complex means this nucleosome contains both DNA and proteins, and this protein, we call this protein as histones protein. These are called structural protein, which pack together to form a chromosome. This histone protein compress the DNA to pack to form a chromosome. Then we can discuss in our figure. Then eukaryotic genome have unique features of exon intron organization of protein coding genes. Exon means that is non-protein protein coding area, and intron means that is non-protein coding area. This representing coding sequence and intervening sequence that represents the functionality of RNA part inside the genome. If we go to the a human haploid cell that consists 23 nuclear chromosome and one mitochondrial chromosome, contains more than 3.2 billion DNA base pairs. As we know, we have 23 nuclear chromosomes and one mitochondrial chromosome present in mitochondria, and these 23 are nucleus specific chromosomes. Then if we go to the structure of this chromatid, uh, this eukaryotic chromosome are usually linear in structure, And a typical chromosome is tens of millions of 100 millions base pair in length, as I previously described as it contains more than 3.2 billion DNA base pairs. And this eukaryotic chromosome occurs in sets. Many species are diploid, which means that somatic cell contains two sets of chromosomes. Each chromosome contains a centromere, that is called centromere, that forms a recognition site for kinetochore proteins, and this yellow-like protein is called centromere region, and this is called one chromatid, and this is called one chromatid. And each then the telomere means end of the chromosome. This telomere contains specialized sequence located at both end of the linear chromosome, and this repetitive sequence at the uh, tail end of the chromosome are commonly found near centromeric and telomeric. This here they call the repeat here in centromeric region we can found the repetitive sequence, and also in telomeric region we can found the repetitive sequence. Then, if we go to the chemical composition of chromatin, this if we go to the this chemical composition of this chromatin, then we can found DNA is 20 to 40%, the most important chemical constituent of the chromatin, and RNA is 5 to 10% associated with chromatin, as rRNA, RNA, all RNA are associated with this chromatin. Then proteins, which proteins? Now we can discuss about the histone proteins. If we discuss about this histone protein, these are very basic proteins and constitute about 60% of the total protein, almost 1:1 ratio with the DNA. And these histones are five types. One is H1, H2A, H2B, H3, and H4. And non-histones, like they are 20% of the total chromatin protein. And these non-histone proteins, such as nucleosome assembly proteins, NAP, and other histone chaperone chromosome protein for remodeling complex, and the other structural protein for holding of this chromosome, that is actin, tubulin, and myosin. This tubulin and myosin and actin are also called as contractile protein for holding of this chromosome. All enzymes, as we know, all enzymes are protein, like we previously described. DNA topoisomer is also enzyme. These are non-histone proteins. The, uh, then we discuss about the chemical composition, that is a chromatin content DNA, RNA, protein, and non-histones. Then, if we go to the structure of this nucleosome complex, then the DNA is wrapped around the histone's core of 8 protein subunit, forming the nucleosome. This is the nucleosome core, 8 histones. This is the core of 8 histones molecules, which wrap around DNA molecules, and this nucleosome is clamped by histone H1. This is the this after wrapping of this gene DNA, and this histone is wrap around the uh, nucleosome, and about 200 base pair of DNA coil around one histone. DNA is coiled around about 200 base pairs of this DNA is coiled around these histones, and this coil on twist so as to generate one negative superton per nucleosome. This coil forms a one negative superton per nucleosome. This is a nucleosome complex. Then, if you go to the proper structure of this octamer complex, then histone tails, different histone tails. If you go to the structure of protein, the protein contain N terminal and C terminal domain, and these histones have different protein modification and protein production. When histone tails are associated with DNA is released for the process of transcription and translation. The DNA tails and these DNA tails are associated with uh, uh, for the process of transcription and translation. If we release this DNA to outside, then transcription and translation will occur inside the cells. Then if we go to the different level of packaging, if we go to the structure of this DNA, this is in 2 nanometer form, this DNA linear structure of DNA. Then it's surround of surrounds the octamer of these histone molecules to form a beads on a string of chromatin, and 11 nanometer in length. Then this in the 11 nanometer in length protein then further processed to form a 13 nanometer chromatin fiber of packed nucleosomes. Then these 13 nanometer fibers form a loop like structures this is called a loop like structures to form a section of chromosome in an extended form. And this uh, a condensed section of chromosome 700 nanometer in length, then packaged into a different form of chromosome, which is 1.4 nanometer in length. How this size is smaller to package the chromosome to fit inside this nucleus. Then if we go to the major difference between prokaryotic genome versus genome, then prokaryotic genomes are small genomes, no specialization. But in eukaryotic genome, because eukaryotic genome is eukaryotes are complex in structure, so they have larger genome and cell specialization, because the genome, uh, the genes are functional in cell specific manners. Like if you go to the stem cells, hematopoietic stem cell, blood cells, muscle cells, liver cells. So these are cell specialization. But prokaryotic do not have any cells. So there is no cell specialization. Then if we go to the genome, then DNA few proteins in simple arrangement, uh, but in eukaryotic genome, that is DNA with many proteins in complex arrangement. As we discussed, the chemical composition of this chromatin, the DNA contains the chromatin contains DNA, RNA, proteins. In proteins, that is histone proteins and non-histone proteins. Then, if you go to the third point, that is contains single set of chromosome, uh, single set of chromosome, and, and it contain one or more, like prokaryotic having one chromosome, but in eukaryotic, as we described, that is 23 pairs of chromosome. Then amount of DNA is smaller because it is small genome and small structure, and but here is larger, typically greater than in bacterial cell. Greater, but in greater than in bacterial cell, they are polycystronic, they are monocystronic. Most of the uh, because polycystronic means there is one origin of replication. The multiple origin, sorry, polycystronic means multiple origin of replication. Here one origin of replication. Then most of the DNA codes for proteins or RNAs, very little junk. There is very little junk DNA are present in prokaryotes, but most of the DNA doesn't code for protein or RNAs. Only 2% of the DNA in eukaryotics are codes for proteins, or 90% are in intron forms. Um, then if you go to the RNA processing are not option for controlling gene expression. Here not RNA processing is not mandatory for gene expression. And then here RNA processing is required because after DNA then it transcribed to RNA, and this RNA is translated to proteins. Then how this RNA is processed, then we can discuss further. Then the mRNA has a short lifespan in minutes, Here the mRNA is long lived because multiple RNA processing occurs. As we know, mRNA contains 5 prime cap and 3 prime polyadenylation, that is poly-A tail. So this is for long lived in eukaryotic genome. This is the complete basics of this genome of prokaryotics as well as eukaryotics. If anyone have doubt, please then.
Sir, what is difference between positive and negative supercoiling?
Yeah, we can discuss about details in further about this organization during this C value paradox about this positive and negative supercoiling. How this coiling is occurs and how positive coiling is stabilize the genome structure in prokaryotics and eukaryotics and in archaea, because archaea sustained in 2000 degrees celsius, 200 to 2000 degrees celsius, how their genome is packaged, how this genome is positive or negative supercoiling, we can discuss it in C value paradox.
Sir, nucleotide
Any question?
always contains two base pair. Sir, what is the difference between a nucleotide and a base pair? That a nucleotide always contains a single base pair, or
Nucleotide contain, suppose nucleotide contains, DNA contains different base pairs. That is called as nucleotides. Nucleotides means A, adenine, guanine, thymine, and cytosine. If A is base pair with T and G is base pair with C, this is called base pairing in DNA. Okay.
suppose, sir, I have 23 nucleotide. I have 23 nucleotide. So, what is the number of base pair will have?
If you have 23 nucleotide, then it is called 11 base pairs. 11 into 2, that is 22. One is there. So 11 base pairs.
Okay, okay, sir. Thank you.
A will base pair with T, G will base pair with C in DNA, but in RNA, A will base pair with U and G will base pair with C. Okay. Then we go further. Then, if you go to the introns, exons, and protein evolution by exon coupling, as you know, DNA, if you go to the structure of this DNA contains different genes, and these DNA different genes are transcribed into messenger RNA, and this messenger RNA coded for translation to form coded for proteins, and these proteins are required for building a organism and their structural and genotype or phenotype. Then if you go to the mRNA structure from this DNA, then DNA to transcribed into this pre-mRNA molecules, and these pre-mRNA molecules having what? Now, these pre-mRNA molecules contain, one is exon part, and this is one intron, and this is called second exon, and this is called second intron, and this is called third exon. This exon is called as protein coding regions, and these introns are called as non-protein coding regions. How this protein is, how these intron and exons are supple to form a proteins. If you go to the splicing mechanism, splicing means these introns are removed to form a exon complex, and this spliceosome machinery cut these intron regions to form exon 1, 2, 3 for protein coding mature mRNA complex, and these introns are removed from the this pre-mRNA, and these exons are shuffled to a mature mRNA. Then how this suppling or exon suppling is occurs? If you go to the for an example in Drosophila, how sex determination is occurs. If you go to the Drosophila double six genes, that is a double six genes pre-mRNA uh, pre-mRNA. This contains uh, 6 exons and 5 introns. How these are coupled to form the male and female. And if you go to the suppling, if 1, 2, 3, 5, and 6 are coupled to form a male specific Drosophila, and if you go to the 1, 2, 3, 4, only this 1, 2, 3, 4, then this is called as female Drosophila. This splicing supple, this exon suppling splicing to form for determination of sex in Drosophila species, because as you know, exon are protein coding region and introns are non-protein coding region. If this suppling occurs only 1, 2, 3, 4, then this species is converted to female. If this exon 1, 2, 3, 5, 6 is supple, and this is converted to a male species of Drosophila. And these are very important splicing mechanism, because these introns are removed and these non-coding regions are removed to form a mature RNA. Then this mature RNA required for translation process to form a proteins, and this is very important process for this DNA. Then DNA converted to this pre-mRNA complex, and this pre-mRNA complex contain both introns and exons, and these introns are removed by spliceosome complex and to form a mature mRNA. And how this exon suppling occurs, and in alternative splicing, this 1, 2, 3, 5, 6 suppling to form a male species of Drosophila, and this splicing means remove of introns. Then if we remove these introns and to form a exon 1, 2, 3, 4 complex, and these are called exon supplings, and these are called intron exon, and these are called intron exon boundaries. Then if you go to the comparative genomics, what is orthologs and what is paralogs? If you go to the comparative genomics, is a field of science in which the genomes of a wide variety of species such as human, mouse, plant, insect, bacteria are studied by comparison with one another, and it involves a comparison of gene location related to gene order, gene loss, duplication, relative gene expression, and horizontal gene transfer. And to use this information to understand evolution and predict gene functions. And this homology analysis, that is identification of similarity in gene and or genome sequence or function, forms the basis of comparison in comparative genomics. Two or more genes with similar nucleotide sequence, two or more genes with similar nucleotide sequence are called homologs. Homologs means what? The two or more genes having similar sequence. This is called homologs, nucleotide sequence. They encode homologous proteins which are similar in amino acid sequence and functions. Homologs may be differentiated into two types. One is orthologs, one is paralogs, and these orthologs, these are homologs which evolve from a common ancestral genes by species. These orthologs are evolved from a common ancestor. They have common one ancestor, and they encode a protein of same function in different species. If you go to the example by comparative genome analysis in yeast, mouse, and human, this is the comparison between yeast, mouse, and human, and one gene is called as MOT L, that is MOT L homolog 1, that is gene is MLH1, and MOT S homologous 2, the gene is MSH2, which encode two crucial protein of DNA mismatch repair system in this species. What is mismatch repair system means? During this replication process, if we go to the mismatch to find out the mismatch nucleotide, such as if A is mismatchly bond formation with C, then this mismatch repair will found by this MLH1 and this MSH2 genes to rectify, because A will pair with only T by 2 hydrogen bonding, and this mismatch repair is recognized by this MLH1 and this MS2 protein, and these genes are orthologous, which have originated from the this mut L and mut S genes of E. coli, and these genes are orthologous to E. coli, and mutation in the MLH1 and MSH2 genes are found in families with a history of hereditary non-polyposis colorectal cancer, that is HNPCC, because if this mismatch repair was mutated, then the colorectal cancer will occur in human beings, and this is very important enzyme, important protein, that is MLH1 and MS2. This recognizes the mismatch repair system in our body during the replication process. Then if we go to the paralogs, then what is paralogs? Now these are homologs which are evolved from a common ancestral genes by duplication followed by subsequent divergence within the same species. This is called these are homologous which evolved from a common ancestral genes by species, that is orthologous. But here these have evolved from a common ancestor, but these genes are duplicated. Like for an example, if you go to the hemoglobin structure, this hemoglobin is contain globin genes, but these globin genes are duplicated, but they have they have, after some speciation, they are converted to alpha globin gene and beta globin gene, and these alpha and beta globin genes are form a hemoglobin structure. The hemoglobin contain two alpha globin gene and also a two beta globin genes, and these alpha globin and beta globin genes, which respectively encode the alpha and beta genes of the hemoglobin, that are examples of this paralogos that is found in human genome. These paralogs are duplicated genes and eventually they are diverged. And this is called paralogos and this is called homologous genes. Anyone have doubt?
ସାର୍ ନମସ୍କାର। ସାର୍ ମୁଁ ଏବେ ଜଏନ୍ କଲି, ଜାଣିପାରିଲିନି ଆପଣ ଏ ଅଲଗା ଗୋଟେ ଲିଙ୍କରେ କ୍ଲାସ କରୁଛନ୍ତି ବୋଲି। ମୁଁ କିଛି ସାର୍
ନା ନା
ବୁଝିନି।
ସେମ୍ ଲିଙ୍କ ରେ ତ କ୍ଲାସ ହଉଛି, ଅଲଗା ଲିଙ୍କ ତ କିଛି ହେଇନି।
ନାଇଁ ବା
Being the
ସେଇ
same
ସେଥିରେ ଆମେ
ଅଛି।
କ୍ଲାସ କରିପାରିଲୁନୁ। ଜଣେ ଭାଇ ପଠେଇଲେ ଏଥିରେ କ୍ଲାସ କଲୁ, ଏବେ ଜଏନ କଲି। ସେ ମୁଁ କିଛି ବୁଝିନି, ଆପଣ ଟିକେ
ଠିକ
ଆଉ
ଅଛି,
ଥରେ
ମୁଁ
ଟିକେ।
ଖାଲି ଗୋଟେ ବ୍ରଶ ଅପ ରେ କହି ଦଉଛି, କ୍ୱିକଲି ରିଭାଇଜ କରି ଦଉଛି। We have
ଆଜ୍ଞା
discussed
ସାର୍।
about the genome of prokaryotic, eukaryotics, and viruses. And if you go to the genome structure, the genome contains a DNA, and these DNA are coiled to form a compact structure that is chromosome, and these chromosomes are located inside the nucleus of the uh, cell, nucleus of the uh, eukaryotic cells, and in prokaryotic cell these are present in nucleoid region. Nucleoid means irregular shape in prokaryotic genome. Then we divided the genome into four categories. One is a prokaryotic genome, eukaryotic genome, organelle genome, and virus genome. And we have discussed about the prokaryotic genome contain double-stranded DNA, and this genome have a circular uh, uh, genome structure, and it contains one chromosome and nucleoid. And in eukaryotic genome, this is double-stranded linear in structure, and this contains many chromosomes. Like example, uh, in human beings, that is 23 chromosomes present, and these chromosomes are present inside the nucleus, but in prokaryotes in nuclear region. And then if we go to the organelle genome, in plants we have found a chloroplast which contains hereditary materials, that is called chloroplast genome. And in mitochondria in eukaryotic cells and also in prokaryotic cells, they have also contain the different kinds of genome. Then if you go to the virus genome, then virus genome contains both single stranded, double stranded, and RNA inside the genome, and they also contain the glycoprotein complex. We have the examples of the SARS-CoV virus, as it contains the crown-like protein above this surface of the genome, and this is called a glycoprotein complex. And these genomes are packaged into chromosome, and these genomes are packaged into this complex structure to form a chromosome. What is the importance of this packaging? This packaging of chromosome is a compact form of DNA, and this DNA is compact form and readily fits inside the cell. This compactness readily fits inside the cell, and it protects the DNA from the damage, and it also protects from DNA from damage. And only packaged DNA can be transmitted efficiently to both daughter cells when cell divides. Then this packaged DNA then divides during this meiosis process. Then we have discussed about the genome organization in prokaryotes. And as we know, prokaryotes contain two different types of genome. One is nuclear genomes and another one is plasmid genomes. And these plasmid genomes are circular genomes, and these are nuclear genomes. And this importance of this plasmid extra chromosomal, uh, this plasmid contains extra chromosomal DNA, and this plasmid provide advantages to bacteria such as antibiotic resistance. This plasmid DNA required for resistance to antibiotic um, in human beings and also in other animals. And in addition, unlike chromosomal DNA, plasmid are often present in many complete copies for cells. Many copies also found inside these bacterial cells. Then if you go to the bacterial content, two types genome. One is bacterial genome and one other one is plasmid genome. Then if you go to the bacterial genome, then bacterial chromosomal DNA usually is circular molecules. That is a few million nucleotide in length, that is 4.6 million base pairs. And this bacterial chromosome contains a few thousands of different genes, and this structural gene sequence account for majority of the bacterial DNA. Then the no transcribed DNA between adjacent genes are termed as intergenic regions, as we described in introns. And then if you go to the packaging of DNA, this circular DNA then form a loop-like structure with the help of this DNA associated proteins to form a looped chromosomal DNA with associated proteins, and this loop contains loop domains. And then this loop chromosomal DNA with associated protein then converted to supercoiling by enzyme DNA topoisomerase, which convert this looped chromosomal DNA to supercoiled and looped supercoiled DNA, or toroidal supercoiled DNA, or electronomic supercoiled DNA by the help of this DNA topoisomerase. And this DNA topoisomerase is having the unwinding, replication, and rewind. If we unwind this supercoiled loop DNA, then will occur. After this replication, then it will rewind of the circular supercoiled bacterial DNA. Then if you go to one example of DNA topoisomerase, that is DNA guidage, which catalyzes the negative supercoiling. This negative and positive supercoiling, as we describe in C value paradox of the circular DNA found in bacteria. Then this DNA uncoiled and relaxed order for DNA polymerase to bind for DNA replication, and the RNA polymerase to bind transcription. If DNA polymerase bind to this DNA, then it will occur, replication will occur to form a increase the copy numbers. If RNA polymerase bind, then transcription will occur. Then we described about the genome organization in eukaryotics. Then we described the eukaryotic genome is a linear and conforms Watson-Crick double helix structure of the DNA molecules, and it contains a nucleosome complex. This nucleosome complex comprising of DNA and proteins structure that pack together to form a chromosome. And this eukaryotic genome have unique features, that is exon intron. This eukaryotic genome also have exon and intron boundaries, and these exons are called protein coding regions, and these introns are called non-protein coding regions. Then a human haploid cell consists of 23 nuclear chromosome and one mitochondrial chromosome, which contains more than 3.2 billion DNA base pairs. Then if we go to the typical chromatid structure, this typical chromatid contains eukaryotic chromosomes, and this eukaryotic chromosome in tens of millions of hundreds are described 3.2 billion DNA base pairs. And these eukaryotic chromosomes occurs in set. Many species are diploid, which means they have somatic cell contain two sets of this like chromosomes, and each chromosome contains a centromere. This yellow part is called centromere, which divides the forms a recognition site for kinetochore complex. This to recognition for kinetochore complex formation. This kinetochore complex, this telomere contains the end part of this chromatid is called telomere, and this telomere contains specialized sequence located at both ends of the linear chromosome, and these repetitive sequence are commonly found in the DNA, and the repetitive sequence are commonly found at the centromeric region as well as telomeric region of the chromatid. Then we have discussed how this chromatin or chromatid uh, complex have chromatin complex have DNA, RNA, proteins, and these proteins as a major part to uh, form a chromosome. And these proteins have both histones and non-histone proteins. And the histone proteins have five types. One is a H1, H2A, H2B, H3, and H4. And these non-histone proteins, they have 20% of total chromatin protein. As we discussed about nucleosome assembly points, structural proteins, which in structural protein which attach during the meiosis process. These structural proteins are called actin, tubulin, and myosin, and all enzymes like you previously described about DNA topoisomerase. And this And the DNA is wrapped around the histone. The red color is the core histone molecules, which contains uh, uh, S2A, S2B, S3, and H4 to form a 2S2A, 2S2B, 2S3, and 2H4 to form a core octamer complex, and this octamer complex is connected with this H1 clamp to form a nucleosome, and in this nucleosome a 200 base pair of DNA coil around one histone, and this coil onto it so as to generate one negative superton per nucleosomes. And if you go to the histone tails, and these histone tails are required for protein modification, protein production, and when histone tails are dissociated with DNA is released for process of transcription and translation. And these tails are dissociated. If we dissociated these histone tails, then this nucleosome is free, and the DNA will unwind to form a replication, transcription, and translation process. Then if you go to the eukaryotic level of DNA packaging, and DNA is at 2 nanometer in length, then it's converted to bits on a string from chromatin to, with the help of this octamer histone complex. Then this 11 nanometer is converted to 13 nanometer chromatin fiber as packaged into nucleosomes, and this section is called extended part of this 30 nanometer fiber, and this is form a loop like structures to form a 300 nanometer chromosome, and this 300 nanometer chromosome again condensed section to form a 7 nanometer chromosome, and this 700 nanometer chromosome this condensed to form a 1400 nanometer chromosome, and this is called the uh, chromosome of eukaryotic genome. This is the complex part. Then we have describe about the difference between prokaryotic genome versus eukaryotic genome. And in prokaryotic genome is a small genome, no specialization, because there is and they have no cells, so there is no specialization of genome. But in eukaryotic genome is larger genome and cell specific specialization, because blood cells only form uh, blood cells have to form only blood cells, not muscle cell or liver cell. So this is very specialized. And the prokaryotic genome have DNA plus few protein in simple arrangement, but DNA with many proteins like non-histones and histone protein to form a complex arrangement, and it contains single set of chromosome. Prokaryotic have single set of chromosome, but eukaryotic contains more set of chromosomes like human beings, 23 pairs of chromosome. In prokaryotic, the DNA amount is smaller because smaller in size. In eukaryotic, that is typical greater than that in bacterial cells, because eukaryotic are complex in structure. They are polycistronic and they are monocistronic DNA, and the most of the DNA codes for proteins, RNA, and very little junk DNA are present. But in eukaryotes, very many proteins, many DNA are junk DNA, and DNA does not code for protein, that is called intronic regions, because they do not code for protein or RNA. And in prokaryotic, RNA processing not an option for controlling gene expression, but here RNA processing allows for several opportunity to regulate the genes. But in prokaryotes, mRNA has a short lifespan because there is no capping or tailing will occurs. But in eukaryotes, the mRNA having capping and tailing. Then we discussed about introns and exons. Introns are called non-coding part of the DNA, and these are called junk DNA. And exons are coding part of the DNA, and this is called protein coding regions. If we go to the DNA structure, DNA are linear structure, and this DNA converted to pre-mRNA, which contains both exon and intron complexes, and these introns are removed by this lysosome machinery, and then this exon shuffling occurs and to form a mature mature mRNA which contains exon 1, 2, and 3. If we go to the different exon shuffling during this Drosophila sex determination, Drosophila contains double six genes in this pre-mRNA, that is, this contains 6 exons and 5 introns, and these 6 exon and 6 exons are shuffled and during this alternative splicing machinery, and this exon 1, 2, 3, 5, and 6 is shuffled to form a male specific mature double six gene mRNA, and this 1, 2, 3, 4 is converted to a female specific mature mRNA, and this determines the sex of this Drosophila by this exon shuffling mechanism. Then we discuss about this comparative genomics, orthologs and paralogs, and we have called 2 or more genes with similar nucleotide sequence. We call this homologs, and these homologs are categorized into 2 types. One is orthologs, one is paralogs. If you go to the orthologs, these are homologs which evolved from a common ancestral gene by speciation. For an example, a comparative genome analysis revealed the budding yeast, mouse, and human. They have a set of genes, that is MLH1 and MSH2, which encode 2 crucial proteins of the DNA mismatch repair system, because it checks the mismatch um, binding of the nucleotides. Like A will bind to only T, not like C. This mismatch repair is guided by this MLH1 and MSH2 genes, and these genes are orthologous, which have originated from this MOT-L and MOT-S gene in E. coli, and mutation of these genes causes colorectal cancer in human beings. If we go to the paralogous, and the paralogous, these are homologous, which have evolved from a common ancestral genes by duplications, followed by subsequent divergence within the same species, and these genes are duplicated. Like if you go to the hemoglobin structure, hemoglobin having globin genes, and these globin genes are duplicated to form a alpha globin and beta globin, and this alpha globin and beta globin encoded by this alpha gene and beta genes of hemoglobin, and this is called paralog structure. Then these are the basics of our genome, how these genomes are organized, and how this genome shuffles occur to produce a mature mRNA. Okay, if anyone have doubts.
ସାର୍ ଆମର ପାରାସାଇଟୋଲୋଜିର ସାର୍ ଲାଷ୍ଟ ବ୍ଲକ୍ 5 ଟା ସାର୍ ବଞ୍ଚିଥିଲା।
ହଁ
ସେଇଟା
ହଁ, ସେଇଟା
ଡିସ୍କସ୍
ମୁଁ
କରିବି।
କଥା ହେଇକି ସାରଙ୍କ ସାଙ୍ଗେ ଗୋଟେ ଇଏ କରିବି, ସେଟା ମନେ ଅଛି ମୋର।
ସାର୍ ଆମର ଯୋଉ ରହିଲା ସାର୍ ଆଡେନାଇନ୍ ଥାଇମିନ୍ ସହିତ 2ଟା ହାଇଡ୍ରୋଜେନ ବଣ୍ଡ କରୁଛି, ଆଉ ଗୁଆନିନ୍ ସାଇଟୋସିନ୍ ସହିତ 3ଟା ହାଇଡ୍ରୋଜେନ ବଣ୍ଡ କରୁଛି। ସାର୍ ଏମିତି କରିବାରେ ସାର୍ କିଛି ସ୍ପେଶାଲିଟି କିଛି ଅଛି କି?
ହଁ, ତାକୁ ଆମେ ଯେତେବେଳେ ବେସ୍ ପେୟାରିଂ ପଢିବ ତମେ, ସେତେବେଳେ ଥାଏ ସବୁ କନଫର୍ମେସନ କେମିତି ବେସ୍ ପେୟାରିଂ ହୁଏ। ଲାଇକ୍ ଯେମିତି ହୁକ୍ସଟିନ୍ ବେସ୍ ପେୟାରିଂ ହେଲା, ନର୍ମାଲ ବେସ୍ ପେୟାରିଂ ହେଲା। ବେସ୍ ପେୟାରିଂ ରେ ହାଇଡ୍ରୋଜେନ ଯୋଉ ଆଉଟସାଇଡ ରେ ଯୋଉଟା ଥିବ, ସିଏ ଓନଲି କ୍ଲୋଜ କନେକ୍ସନ ୱିଥ adenine, କ୍ଲୋଜ କନେକ୍ସନ ୱିଥ ଦ thiamine, ସିଏ ସେଇଠି କେବଳ ହିଁ ତାର କ୍ଲୋଜ କନେକ୍ସନ ରେ ବଣ୍ଡ ଫର୍ମେସନ କରିପାରିବ, ନଟ୍ ସାଇଟୋସାଇନ୍ ସହିତ କରିବ, କାରଣ ସାଇଟୋସାଇନ୍ ଫ୍ରି 3ଟା ଥିବ ଫର ହାଇଡ୍ରୋଜେନ ରିସିଭର, ବଟ୍ ଇନ୍ adenine only 2ଟା ଥିବ, that is detected by thiamine, not cytosine or guanine. ଆଉ କାର କଣ ଅଛି?
No sir, ଆପଣ next page को next minute को ଯାଆନ୍ତୁ।
ଆଜି ପାଇଁ ଆମେ ଏତିକିରେ କ୍ଲୋଜ କରିବା ବେସିକ୍ସ ରେ। ନେକ୍ସଟ ହିଁ ମୁଁ ସବୁ ତାର ନେକ୍ସଟ ଆପ୍ଲାଏଡ ହିଁ ପଢେଇବି। ଆଉ କାହାର ଯଦି ଡାଉଟ ଅଛି ବେସିକ୍ସ ରେ ପଚାରି ଦିଅନ୍ତୁ। କାରଣ ନହେଲେ ନେକ୍ସଟ ଗୁଡାକ ଯେତେ ସବୁ ଅଛି ସବୁ ଆପ୍ଲିକେସନ ଅଛି। ଆପ୍ଲିକେସନ ଟୁ ଦିସ୍ ଜିନୋମିକ୍ସ ଆଣ୍ଡ ପ୍ରୋଟିଓମିକ୍ସ। ଆଉ କାହାର ଯଦି ଡାଉଟ ଅଛି ପଚାରନ୍ତୁ। ଠିକ ଅଛି, ଆମେ ଆଜି କ୍ଲାସ ଏଠି କ୍ଲୋଜ କରିବା। ନେକ୍ସଟ କ୍ଲାସ ରେ we can discuss the application of this genome and genomes. Okay.
Yes, sir.
ओके सर।
अरे जो पीपीटी ଟା सर शेयर कर दो।
हाँ हाँ मैं शेयर कर देंगे। थैंक यू।
थैंक यू सर।
थैंक यू सो मच।
हेलो आम असाइनमेंट कोठी सबमिट करबा कथा कि जानिछन्ति कि